Brings me then to announcing the first speaker, Jean-Jacques Garaud. Jean-Jacques had a, I suppose, incredible career in what some people may describe as something like this, but very much in the head of pharma research and early development at Roche, chief medical officer at Roche, then started with something that was, of course, also already happening when these animals were walking around. New life forms were appearing, which were probably laughed at by the dinosaurs because they were small, rather useless mammals. But the mammals made it and the dinosaurs didn't. So he's been in all these places in venture capital. And so, Jean-Jacques, very welcome to Leiden, and we'd like to hear your thoughts about these new life forms.
Thank you. Thank you. Good afternoon. Thank you, Adam. It's quite an honor to be here in such good company. And may I have the first slide, or? It's here. It's only here. Okay. Do you see the slides? Okay, good. Let's go back. What I am here for is to actually spend some time sharing my thoughts and my experience and my learnings about moving from big pharma into a smaller world, but which is actually a big world, and give my reflections about the emergence of new life in the healthcare industry. When I left Roche 5 years ago now, I decided to get into the interface between the academic world and technology transfer offices, venture capital, trying to be helpful to the transformation or industrialization of great technologies and a great knowledge in some biological pathway in a given disease, etc. Basically helping scientists from the academia to transform their discovery into an actual innovation, and that would bring added value to public health and to society, which is basically the end game of what we are doing every day. So I actually did some research and tried to understand what was the meaning of emergence of life, what we call biogenesis. And it goes back 3.7 billion years ago. And this happened in what we call now the primordial soup. And for the primordial soup to actually generate life, you not only do have to have the right condition, external condition, but you also have to have a convergence of those right external conditions with the presence of key building blocks. If you don't have the two together at the same time in the same place, it just doesn't happen. And for the emergence of life on planet Earth for the first time, that happened on the shorelines of the ocean and at the bottom, deep bottom of the ocean in the vents. And this is one example. These are the stromatolites. And these are concretions of that happened, created by cyanobacterium, the first living organism that created the concretion layer by layer, and we still have the evidence of that in the shorelines of Australia, if I remember. So the concept here is the primordial soup and the right building blocks at the same place at the same time. Now, if you look at the biogenesis of what we call today big pharma, what is interesting to look at is try to identify what were at that time the building blocks, what was at that time the great environment that was helping out, making it happen. And big pharma has established itself on a certain number of key disciplines: medicinal chemistry and biochemistry, pharmacology, physiology, clinical pharmacology, toxicology, regulatory affairs because it became more and more a regulated environment, and also what I call intelligent marketing or marketing science. Those together were really the pillars of the success of big pharma from the late 40s to the 2000s, let's say. And at that time, in order to build the pillars, to build those functions, and for them to reach excellence, it meant that everything was built in-house, brick and mortars. internal resources and very little outward reaching out. Everything was, all those disciplines were built into those large companies, and they were responsible for the success of those companies. And by working together, they established a certain process. They had a setup that became extremely successful. But for that setup to be successful, it had also to respect a certain discipline around the process, which means also a certain rigidity. And it's almost an understatement. And one of the rigidities that I have discovered later in my career when I became the head of research at Roche was actually to see that most of the budget of a research organization in big pharma was actually captured by the payroll for the researchers and the buildings and the tools and the material, and very little of that money, which means most of 95% of the money was for internal resources. Very little of that money, only 5%, was available for trying to play outside of the big pharma campuses, work with biotech companies, work with academic centers that may have innovative approaches. And it was not possible because we didn't have the resources for that. We had billions of dollars, but we didn't have the resources to do something different. And that was at the origin, in part, for the laying off of a lot of people in research and development in big pharma, so that a certain amount of the portion of money that was devoted to external innovation became more important and more available. This is something that big pharma realized relatively late, but did realize. Meanwhile, there was a major revolution in lab science that occurred during more or less the same period, starting more in the 60s and becoming more, I am losing my microphone, it's better, and becoming more and more impactful in the 70s and 80s and 90s. And this is the creation, these are the right conditions for creating a new primordial soup. And that led to the creation of a certain number of new building blocks. The new technologies led to new therapeutic modalities, new tools. The computing power became exponentially growing and was helping a lot for handling big data sets, bigger and bigger data sets. And it led to the concept of translational research and translational medicine, which is the relabeling of something we used to call experimental medicine, but with the new tools. And that led to also the concept of using biomarkers in analyzing the pharmacological activity of a potential product. And that led also to realizing that one actually can try to select the patient population that is most likely to benefit from a given treatment, and we call that personalized health care, by using the potential biomarkers that may actually select in or out the patient population that may benefit or may not benefit from a treatment. So this is what happened during the same period, and during that period also, these are the building blocks, but the external environment did change as well. It's better? I have a new friend. We have, what did I say? Yes, the external environment changed, and during that period, large amounts, fairly large amount of public funding became more and more available, like the cancer plan launched by President Nixon in the US, but also private equity money coming from venture capital was also becoming more and more available, and is today more available. And at the same time, a new breed of entrepreneurs were actually coming from the academic world, as well as from pharma and other places, mainly from the academic world and pharma that was starting to shed senior leaders from their companies. And at the lower level, there was a huge shedding as well, as I said, from big pharma, and that expanded the pool of available talents to actually go and feed the need that the nascent biotech industry was actually generating here. So this is what is happening at the same time as the big pharma is getting rigid. and sclerotic. So talking about dinosaurs and species that have disappeared, this is a small list of relatively big pharma companies from the 70s, 80s, and 90s that today have disappeared into a merger, into the meanders of mergers and acquisitions. Because one of the things that actually big pharma did was for them to acquire larger companies, to acquire another company to become larger, to generate economy of scale, to shed people, to improve, if you will, the marketing of the existing products, gain some new products into the pipeline. But this is not the best way to be innovative, quite frankly. This is maybe a way of being innovative from the pure financing standpoint and from the pure capitalistic standpoint, but it just doesn't mean that you are actually generating new innovations, new ways of doing things. And most of the activity of those R&D groups were actually to integrate with each other and not necessarily to look at what is happening in the outside world. I have worked in my last 30 years as well in the industry for 4 of those companies, and they are gone today. But we have actually had good successes in some drugs. Now, I am going to take, after those generalities, to give you a few examples of what I have done over the last 5 years by helping academic groups to try creating their companies. And over the last 5 years, when I left Roche, I started working with the technology transfer office of INSERM, and which is called INSERM Transfer. And what happened there is that I became their advisor to give them, to the PIs and the young scientists some guidance as to how to prepare their plan, how to prepare their presentation to potential investors, and so on. And I have participated to the creation of 6 of those companies coming from those days. And I can say we have been successful because those 6 companies are still around, so actually this is a good start. This is one of them. This is Enyo Pharma that was created a few years ago. And the story is the following. It's about meeting with the people. And INSERM Transfer asked me to meet with the PIs in Lyon. These are virologists who have actually created and curated a pretty smart database of the interactome between the viral proteins and the human proteins that are targeted by the viruses to actually capture the human machinery to their advantage and survival for them to replicate and be pathogenic sometimes. So that's, in that case, what they have been able to do is that they have identified the hotspot of the protein-to-protein interaction, and they have also identified the peptide sequence that was from the protein, from the viral protein that was responsible for the interaction with the active site on the human protein, so that you have here the identification of the pharmacophore. And this is a good place to start for a medicinal chemistry work with a good structure-activity relationship approach. And that's a way of doing. Plus, these people have generated approximately 300 peptides, such peptides, on different protein-to-protein interactions, so they have the tools to be used experimentally in vivo, in vitro assays. In addition to that, there is another approach, which is the reverse one. You know the protein that is targeted, you know the site of the protein that is targeted, therefore you can actually identify the drugs that are available in the pharmacopoeia, and that actually can intervene there on those proteins. And if you need, you can actually possibly have access to that drug, and I will talk about that later. So it's a very smart approach here and a very innovative approach here. The targets are not the viral proteins anymore, but the human proteins. So their work has lasted for 10 years almost. They have burnt public funding, one important building block. They have burnt public funding approximately between 7 and 10 million from Europe and from the French system. But they have created, out of 80,000 articles, they have selected 88,000 papers, and they have actually identified 17,000 interactions between the viral protein and human protein. Sometimes some of them are overlapping, and they have generated a golden library of 300 peptides that are tools and are active peptides, pharmacologically active peptides. So what was their question at the time when I met with them? Because they had the tools, they had actually all the methodology, but they were kind of running into circles for a couple of years without knowing what to do next and how to integrate this information in order to make something that could be valuable to public health and create new antiviral drugs. And the issue was that they were not able to focus. They didn't know how to organize their setup and how to organize their knowledge and tools in order to present something that could be valuable to investors. So we realized that they had actually a good intellectual property position in influenza and hepatitis B, and we focused on that, and we forced them to focus on that, so that we created a company based on that. And the two assets were very clear, very readable to the investors, and particularly in hepatitis B. We were able also to identify a molecule that was sitting on the shelf of a small biotech company in France, and we were able to acquire for little money that molecule, and today this molecule is in phase 1, completing phase 1, and going soon into the patients before the end of the year. And that molecule is targeting FXR, and this is an FXR agonist. FXR agonists are known today for something else, which is NASH syndrome, but here we're looking at hepatitis B, because the hepatitis B virus is following that path to actually capture the metabolism of the hepatocytes to replicate itself, as I said before. So viruses are great revelators of network pharmacology, basically. This is a good learning position here. So here you have a brief history of this company, and what were the building blocks? Good science, world-class science, quite frankly. Good patronage from the technology transfer office at the time. They had the intuition, didn't know how to move, but they had the intuition. Were able to find a great CEO to sort this out. Were able to spend time with them and sort out the key elements that they had in their hands so that we could actually provide a value proposition, and we had good connections around with investors to make it happen. So the human factor is here very important. It's about connecting the dots, identifying the gaps, asking the right questions, and being able to answer those and identify the optimal value-adding pathway toward the creation of a company. The second program is very dear to me because this is the company I'm running today called Inotrem. So I am a young CEO, as you can see. And here, again, Insert Transfer asked me to go and visit people in Nancy, where they had identified the role of the TREM1 receptor in critical care setting, in acute inflammation in critical care, particularly with the possibility of using the peptide they had isolated as a potential therapy in septic shock. My first reaction, having spent 30 years in pharma, was Vaderetro Satanas, don't talk to me about septic shock. But when I do that, I still go and I'm always very curious and listen to people. And I was quite impressed with the quality of the biology and the work they have done preclinically to demonstrate the activity of their potential product. And here is an example. First of all, I have to talk about the TREM pathway. The TREM1 pathway is it's a protein that is a receptor on myeloid cells, and that is associated mainly to the toll-like receptors and also to the node-like receptors. And when the toll-like receptors are activated, the TREM1 pathway gets activated and creates an amplification loop for the innate immune response. And what it does is that it's a maintenance loop. And in some people, that loop, that system gets into an overdrive. It's like that the inflammatory response, immune response, is excessive compared to what was the initial insult and is driving the patient into shock and potentially into death. So it's a pretty severe condition, and there is no specific mechanism-based therapy for that. So what we have identified here is a peptide that actually can moderate this overdrive and bring it back to a normal behavior. What you see in black is the placebo group in the animal model. In the mouse model, this is not necessarily the most relevant, but it is a model that has been used broadly. And the orange bars and dots show the activity of the product on the left-hand side on the pro-inflammatory cytokines and these anti-inflammatory cytokines, that is IL-10. And I must say that 20 years ago, I've worked with Sander van de Venter on IL-10, trying to find an indication for that product. And we, at that time, badly failed, didn't we? But it was a lot of fun. On the right-hand side, you see the effect of the product on survival. So in the orange curves show that the mouse are surviving nicely, even if the product is administered 24 hours after the injury. That actually is an indirect way of saying that here you do actually have an effect on the maintenance loop, not on the triggering event. So it's quite interesting. This is one of the things that really turned me on when I listened to those guys. And the last one in the middle is actually showing that the bacterial clearance is improved. There is no antibiotic here. That peptide doesn't have antibacterial activity. But what it does in restoring a normal immune response, particularly cellular immune response, there is better clearance of the bacteria from the blood and the spleen. And if you use, for instance, an anti-TNF antibody here, what you would see is the opposite, a worsening of the bacterial clearance. So all those points, because my background is in critical care medicine, I omitted to say, and I am an infectious disease specialist and a microbiologist, so I have worked a lot in that. All those points were triggering a lot of intriguing questions to me, and I followed up with them, and I helped them building new models that were actually closer to the human model, which means creating a septic shock in the mini pig. What we did here, this is like an intensive care unit. This is at the School of Surgery in Nancy, which is actually a pretty famous one. What we did was actually creating a peritonitis in the mini pig, inoculating the feces of the mini pig in the peritoneum, and a few hours later the mini pig starts to be sick. It is intubated, mechanically ventilated. We fill them with fluids, and then when it gets worse, we actually treat them with vasopressors. So, and of course, it was blinded. We have an intensivist that was blinded to the study, and he was taking care of the mini pigs according to a predefined protocol. And what we see here on the right-hand side, that there is an impact on mortality. There is no dead pig at the end of the 24 hours, and half of them are dead on the other side. On the left-hand side, we actually look at the blood pressure, and you see that despite fluids and then vasopressors and increasing doses of vasopressors, you have the blood pressure keeping going down, whereas it is maintained to something that is acceptable with the product. But more interesting is that you see the use of vasopressors, and it is much lower use of vasopressors with the product, which means that there is a conservation of the system here, and a way to potentially have an impact on shock and hence mortality. So the questions that those people were asking themselves at the time with this type of data is, how do we develop a drug now that we have a good evidence of the activity of the product? And again, this is the same answer. We don't know, we don't have the knowledge, we don't have the right people around us. And even if we have people who tell us they know how to do it, they are not necessarily trustworthy. And we need to have the right support. And at the same time, although the investors were actually seduced with the quality of the data and the quality of the biology, they didn't want to invest if they didn't have someone who had actually the right background in drug development. This is where I accepted to become the CEO of that company, and this is how we were able to raise 18 million in the beginning of 2014, so that we can take the product up to the end of early phase 2. We are starting the phase 2 very soon in Belgium and in Netherlands. We have 2 centers in Netherlands, by the way. And beginning of June, we'll be able to see the first patients treated with that product. And what is interesting, the man who invented the product, Professor Sébastien Gibaud in Nancy, is also a critical care physician and has a center. And it's one of those rare events where a physician can administer to his patients a product that he has discovered. And here again, the human factor was important. All the building blocks were there, if you will, good science, good IP, good ideas about actually developing the drug as a personalized healthcare, because we have a good knowledge of the biomarkers, mechanism-based biomarkers, where we can actually select the patients in for those patients who only have the TREM pathway activated. So with that, we believe that we can make a difference at this point in time. So in that experience, in order to start concluding, what was the greatest hurdle that people are meeting when it comes to transforming discovery, good science, into innovation, as I said, something that brings added value to society? Well, this is actually access to the right knowledge at the right time. This is access to the know-how, not only the knowledge, but the know-how. And what I have seen, and I was really shocked by that, is that the knowledge is highly fragmented. You really have to spend a lot of time to try to identify, if you are a naive individual in this field, to identify the knowledge you need to know, what you need to know, and how you need to actually execute. And it's almost impossible. This is, again, where the human factor is the most important. You may have the right soup around, in primordial soup. You might have all the right building blocks, but what is missing is the chef with the brigade in the kitchen to be able to do the right thing and to create something very specific. This is like haute couture. This is not prêt-à-porter. There is no preset recipe. You just have to be creative each time you are facing a new situation. So this is what I had to share with you today, and I hope this is of interest to you. One last side, as a little wink here. We are talking in France about the disparition of all species and the birth of new political animals, and I hope that will refresh the atmosphere a little bit here in Europe. Thank you.